Bom dia, pessoal. Espero que estejam todos bem. Novamente iniciando mais um dia com as nossas atividades. Hoje nós teremos o prazer de receber o professor Dr. Thomas Leya. O professor Thomas Leya, ele é biólogo, né? Ele trabalha com algas, ele fez o doutorado dele na Humboldt-Universität de Berlim. Atualmente, ele trabalha também ainda na Alemanha, no Fraunhofer Institute, e ele trabalha dentro do instituto, ele trabalha numa coleção, ele é curador de uma coleção de algas criofílicas, né? De algas que crescem sobre o gelo, certo? Dentro desse instituto também, ele é líder do grupo de pesquisa de algas extremófilas, estudando então biologia dessas algas, Permafrost, né? E principalmente algas que são coletadas em ambientes polares e em habitats de grande altitude dos Alpes, né? Trabalha também com taxonomia, filogenia, fisiologia, biotecnologia ligada à questão molecular, né? Desses tipos de organismos de algas extremófilas. E ele vai estar tratando hoje com a gente sobre esses grupos que ocorrem nesse tipo de habitat e o potencial desses grupos na pesquisa, né, na biotecnologia. Então, eu vou chamar o professor aqui. Gostaria de agradecer desde já a participação dele. Hello, Thomas.
Hi.
Thank you so much for stay here with us and share your area, your research with us. You have the words to start now and talk with the people.
Thank you, Valentin, for the nice introduction and having or giving me the opportunity to introduce this very interesting field of snow algae to you all. It's a very special group, and I will share my screen now. I hope it will work that we tried it out. So you should see my title page now. I hope that works.
Yeah.
Yeah, exactly. And we decided with Valentin, if you have questions, you could write them in the chat box. And if Valentin feels there are enough questions, then we shortly interrupt my talk and I can answer the questions. So I will give you some insight into this group of extremophilic algae, the snow algae, a very broad term, basically describing algae and others living on snow and the surrounding areas in polar and high alpine regions. I will give you a lot of basic information first. So this overview shows you where we're going in the next 3 hours. My laser pointer here. So first, I will tell you what actually are snow algae and what is this term psychophilic or cryophilic? What does it mean? What makes the algae so unusual? So a lot of basic information from there. And the next part will be that we will go into the field. I will show you a lot of pictures from where we sample our algae during expeditions, and then how long it takes basically to establish cultures in the lab so that other people in their lab can work on these cold adapted algae under lab conditions. It's always a difference if you do field experiments or lab experiments, of course. And then we have a break in between here. And the second part, then we'll go into the products of algae, because here at Fraunhofer, we actually develop industrial products. That's our job at the Fraunhofer Society. We do basic research if necessary, but the dominant part of our work is applied research. So I will tell you what products we can find in those snow algae or cold adapted algae, what special products we don't find in other algae. You know, a lot of other algae, which of course also produce pigments and so on. But what is a special thing about the snow algae to use in products? And at the very end, very briefly, I can introduce you to one product we have on the world market since more than 10 years already. So the first part, I will show you a lot of information about the basic things about you have to know about snow algae and cryophilic algae. It all started basically with a red dot. In 1818, so it's more than 200 years ago, there was a voyage from some English scientists and seafarers, and they went to the northwestern tip of Greenland. And what they found there, observed there, was these so-called crimson cliffs. You must imagine the very northwestern tip of Greenland, really. And suddenly you always expect white snow, and suddenly there was this red snow. And of course, the Captain Ross, sent out some of his colleagues to collect some samples, and they came back, and they in the sample they observed these red dots. They didn't really know what it was. They carried it home, and at home in England, actually a lot of scientists heard about this miracle of this red cliffs, and they all received samples of these red dots, red little globules, and some of them put them into water, into culture medium, and in some samples these green flagellated cells resembling Chlamydomonas reinhardii appeared, and then people classified it on this basis and said, okay, it's Chlamydomonas nivalis. Chlamydomonas, some of you might know, this genus of microalgae is a little egg, basically a green egg with little two flagella, and with the flagella the algae can swim around. And it's a very big group of 200, 300 species, and it's all being reviewed now. It's being split up, but at that time it was Chlamydomonas nivalis because the epithet of the species nivalis says it's coming from snow. That was basically the knowledge and what people assumed over the last 150 years, I must say. In the last century, people looked a bit more closer, and they found that actually the life cycles of these flagellated cells, these Chlamydomonas or Chloromonas cells here in the middle, this green egg with those two flagella, it's very diverse. It can multiply asexually just by splitting up into 2 cells or 4 cells, releasing these vegetative cells. They look all the same in the asexual reproduction cycle. They can reproduce sexually as well. Then the cells divide with meiosis, make little gametes. They fuse again. The gametes look like the adult cells, and the zygotes, they carry 4 flagella, but they also form red cysts or yellow cysts, and they germinate and look the same as before. Then you have side cycles where the vegetative cells can form thick-walled planospores, so an asexual cycle of a resting stage like the cyst, or they can get palmelloid stages, meaning they have a gelatinous matter around the cells, also protecting them from environmental stress. So they can appear in very diverse, different types of cell appearances. Of course, also these red cysts which were sampled on crimson cliffs. So people thought it's all okay, it's still Chlamydomonas nivalis, and we're happy with it. The problem was that actually these days we believe that at that time in 1880, when people put these red cysts into water and they believed that the flagellated cells germinated out of the red cysts, that was not actually true, because in a snow you find a big diversity of other Chlamydomonases. I show you here an 18S tree, a phylogenetic tree I made during my PhD thesis 16, 17 years back. And at that time, I already found out that in snow, if you sample, you get plentiful of Chloromonas and Chlamydomonas species. All the blue ones here in the tree are from Spitsbergen, from polar snow or alpine snow. Basically, this is all polar snow, and you see there are like 15 different species of Chlamydomonas or Chloromonas like algae. Also, these red cysts occur here, but also the orange and red cysts are also occurring in different other clades. So compared to what people thought before, having only one species, it actually, the true story is in snow you find many, many more different Chloromonas and Chlamydomonas species some of them forming red cysts, some of them forming orange cysts, others forming yellow cysts, some surviving winter in their green form, or as gliocysts, also cysts with a thick gelatinous layer around. So there's a high diversity within a snowfield, and not only one species. I said already before that this big group of Chlamydomonas or Chloromonas, because that's the two genera people assumed representing these green eggs with the two flagella, is now being split up with the new molecular methods coming up. The whole 200, 300 species are being split up into different genera, and that also happened with this red clade here of the red cyst, because we knew it was very separate from all the other clades, and we last year and this year or last year actually, we investigated in a more intense study in 2019 already, and we analyzed red cysts from all around the world, from Alaska and South America, I think wasn't in there, unluckily, but from Asia and Europe and Antarctica. And we described the genus for the red snow with the new genus Sanguina, with two species being the dominant red cysts and orange cysts on snow. There are some other genera involved, but actually what used to be Chlamydomonas nivalis basically is Sanguina nivaloides now, and Sanguina aurantia. This is a free available research article, so you're happy to download it. As I said earlier, nobody actually made it to germinate the red cysts. Remarkably, a group in Canada, in the Quamby lab at the Simon Fraser University in Vancouver, they claim to have germinated red cysts now, and they are in the way of publishing it. That would be a real breakthrough, that people can really work with living cultures in the lab of this red snow. All the cultures we have here in our collection are these isolates from the other Chloromonas and Chlamydomonas, which are also Psychophilic and Cryophilic and Cryotrophic from snow, but not they don't represent this clade of the red snow, now called Sanguina. So what are permafrost, or what are snow algae? What makes them so special? We have two conditions in polar regions. We have a summer condition where the snow fields or the area there consists, of course, of soil or rock substrate, and it's covered with glacier ice or snow. And then in summer, we have a temperature around 0 degrees in the snow, so it's kind of half melted. We've got slow slush and release of nutrients. Salts are usually very low because you have freshwater rain being basically aqua destillata raining on the snow. Also, the snow basically doesn't consist of any nutrients. Phosphorus and nitrate is always below detection limit. You've got, as I said, rain going on the snowfield. So somehow the mass development is very astonishing. With this very low nutrients, I'll show you pictures later on, that these mass developments of green snow and red snow can develop on these fresh snow fields and glacier fields, which can be really immense. It's amazing. The soil itself can warm up to 15 degrees, while the snow, of course, is around 0 degrees. Winter then is a completely different situation, with becoming winter in September, October, and so on. Temperatures go below the freezing point of water, below 0 degrees. We have got a concentration of salts because only the water freezes, and all the salts which are available, of course, from the dying cells and so on, is a high increase of salts. So we've got osmotic stress on these algae. Water is also evaporating, so we have a kind of desiccation process there on. And you see a lot of the Chlamydomonas cells, the green flagellated cells, they form the red cysts or orange cysts or yellow cysts, different groups we find on the snow really when we sample it in early summer, where it's the same late winter condition, or in spring. Some green algae, like this long filaments here, Rhaphidonemma, they obviously freeze in the stages and they survive the winter. The soil algae, which are green in summer here on the left, they also in the winter become orange. They accumulate carotenoids, they make thick walls and survive on the soil also on the snow sometimes. And this is a condition where the snow falls on the whole snow. Permafrost algae covers them in 2-3 meters thick. And we measure the temperature below that. And during winter, we've got -7 degrees here. And that's obviously the time of the temperature, the conditions in which the algae overwinter. And in summer or in spring, the same starts all over. With the rising temperatures, the snow melts and releases the nutrients and so on. The life cycle about the snow algae is not really known because nobody really could use the red cysts and have the trophic stages, the multiplying stages, to really to investigate the life cycle. But we are on the way, obviously. As I said, the temperature is a decisive stress for the algae because temperature rising releases salts. If it's the temperature decrease, it's freezing, and then you've got hyperosmotic stress or hypoosmotic stress, and of course the temperature stress itself. This is annually and also diurnally because you have freeze and thaw cycles during the day sometimes, depending on the weather. But of course they have light stress and nutrient stress on a snow field. In the polar regions you have in summer 24 hours of light, and in winter of course also a few months, 3 or 4 months with total darkness. This is of course stressful for a plant as well. And you have a lot of other environmental factors determining the occurrence actually of the algae, because we always ask ourselves, where do this red snow and why does it occur at certain patches in the alpine areas, also in the Andean mountains, for example, in Ecuador, in Colombia, on Mount Pinatubo, for example. But why does it occur there and not on another alpine region? Now, obviously, geology and topology of the snow field with the snow chemistry and the snow wetness play an important role. Here, as I said, we study the adaptation mechanisms in the first site because we have to do this basic research, and then we transfer that knowledge into industrial products. Coming to the term of cryophily or psychophilic snow, what does it mean? Psychophilic, cryophilic. Cryophilic and psychophilic is just, one is Greek and one is Latin, saying just cold loving. So the algae love the cold, but we have to differ between algae which are obligately adapted to the cold temperatures, showing these two growth curves. This are curves of the specific growth rate, depending on the temperature. So with a rising temperature, the growth curves changes, of course. The orange curve, for example, is very easy for Chlamydomonas reinhardii. It doesn't grow below 12 degrees usually, so that's when the growth rate starts becoming positive. And with rising temperature, the faster Chlamydomonas reinhardii grows, to a certain degree, of course. The cryotrophic algae, they, not unlike reinhardii, they actually can grow at low temperatures, but actually prefer higher temperatures as well. So they have low growth rates at low temperatures at 2, 3, 4 degrees, and then with higher temperatures also the growth rates increase. With the cryophilic and the psychophilic algae, it's different. They have have growth rates which are high and highest, and at the optimum below 15 or 12 or 10 degrees, depending on how well they are adapted as. There's a species specific, but with a rising temperature, then the growth rate decreases again, and at a certain temperature, the algae actually die. This can be at 8 degrees, 10 degrees, 12 degrees. That depends also on the species. So that's a big difference. The cryophilic, psychophilic algae, they die at higher temperatures and have growth rates which are high at low temperatures, and the mesophilic or the cryotrophic ones have high growth rates at higher temperatures and won't usually grow well at low temperatures. So that you have to have in mind to have a difference if you look at products, because if you want to produce something in a cold adapted algae, that's always advantages at low temperatures only, because at high temperatures the algae can be grown better, but these obviously won't grow at low temperatures if a snow alga produces a product which is not being produced by a mesophilic alga. So the threshold is, according to Morris, I'll show you in a second, around 50 degrees. He reviewed a lot of information on cryophily and so on, and he determined basically if the optimum growth temperature is below 15 degrees, then we can talk of a psychophilic bacteria he was working on, but that can be transferred to algae or macroalgae as well. This is a typical growth curve of a psychophilic alga of our strain number 50. A student who did his bachelor thesis in my lab just this month in October, he finishes it. And this is a typical growth curve. If you look at the cell concentration per milliliter along the time scale, then you see with a culture growing, the cell concentration rises if you culture the algae at 2 degrees. If you culture the same culture starting at the same cell concentration at 12 degrees, then it's kind of replicates a little bit, but not really, and then slowly you go into the stationary phase, so the culture really doesn't grow, and then finally it will die. So for this species and for this strain, the 12 degrees is already more than the threshold, and I know the maximum is of the growth rate is around 8 degrees or 5 degrees. And that alga actually we isolated. It's like a chlamydomonas. It also usually have 2 flagella, which it throws away very easily. But it looks like a typical chlamydomonas cell, and it has been isolated from this dirty red snow. So within this red snow, the red one is really sanguina, but we grew these green algae out of this red snow we sampled. That's a typical mistake the people in 1880 made. What comes out of the red snow does not necessarily represent the red snow. This is really a green snow. I can show you a bit later. Another example for this cold adaptation, if you compare chlorella isolates, one isolated from Hamburg in northern Germany in a temperate area, and one isolate from Antarctica. And colleagues in Hamburg, they investigated the photosynthetic activity with a PAM apparatus. And if you measure the PAM photosynthetic activity, the ETR rate at 5 degrees of the both isolates, you see that the isolate from Hamburg is not so efficient in using the light, and also with higher intensities, here are the light intensities, the efficiency of the photosynthetic apparatus goes down. Whereas the cryophilic, or at least the isolate from Antarctica, it's cold adapted, I would say, it's cryotrophic rather than cryophilic, but it's cold adapted, and it has a high efficiency at low temperature, at low irradiation rates already, and also it can cope with higher irradiation rates. And this is very typical, obviously. They can cope with a wide range of conditions, with low light, with high light, and with the temperature in a certain range, but the cold adapted strains and also the chlamydomonas strains, they can grow very well at low irradiation levels, but also can cope with high irradiation. They have protective mechanisms against that, so they are well adapted to that special environment in the polar regions. Coming back to this 50-degree threshold, if you want to transform it a little bit into the organisms, one can say below 15 degrees, the optimum temperature can differ between 0 to 8 or 0 to 10 or 0 to 15 degrees, depending on the species. Then we talk of psychophilic, obligate cryophilic, snow algae or permafrost algae, represented by these typical genera and species, Chloromonas, Remiazii, Chlamydomonas, Clinobasis, or Rhaphidonemma. They are all psychophilic with an optimum between below 15 degrees. If you go a bit higher, then we have also other soil algae. We have also some fungi we find on the snowfields. Staphylococcus is everywhere, but it shows to be cryotrophic. So it can grow below 50 degrees, but it's not very happy there. And it likes a bit more higher temperatures. So it's kind of intermediate in between. And then, of course, you find a lot of cyanobacteria and also mosses and so on. And they all prefer higher degrees. They are not psychophilic. They are cryotrophic. They obviously survive there on a snowfield, but they actually like higher temperatures. And here on the right, this is a paper by Morita from 1975, quite old. And he says, reviewing a lot of papers on cryophily and bacteria and so on, he said, by use of the definition employed in this review, a start can be made to end the confusion concerning the term. Because it's really confusing. People don't really know what is the threshold for psychophily or cryophily. And this threshold of 15 degrees, in my experience over the last 20 years working on snow algae is a good measure to differentiate between cryophilic and cryotrophic algae. And especially if an alga dies at higher temperatures, around 15 degrees, then you can be sure you have a really cold-adapted, strongly cold-adapted snow algae. Otherwise, you're working with a microalgae. That's fine, but it's not the special about snow algae. So that was the introduction, basically, and all the background. I don't know, well, are there any questions already coming up, or?
We have a question. One question, actually.
I can just, yeah, I can stop my presentation and.
Yeah, and then we can see.
Don't know yet. So Gabriela asks, so basically the red snow is a cyst and we don't know yet how it would be in its germinated form, right? Exactly. Gabriela, that's exactly true. A lot of people don't understand it because they always sample red snow from a snowfield. You find it, if you know what you're looking for, it's easy to recognize it. When you don't know it, you think it's dirt. But people who put a microscope up and look at the red snow and they see these red globules and they bring it home, and there will always something be germinating out. If it's not Staphylococcus, I also made this mistake once. I plated red snow, even single cysts from a snow sample, into micro well plates, like, you know, this 96 well plates. And in one well, I thought I put one cyst only, one red cyst. And suddenly this well was green. I was happy because I thought, whoa, and I have a germinating red snow now, a culture. And then I looked in, and luckily it was Psychococcus. Psychococcus is so easily to recognize and to distinguish from the red cyst, what I assumed. So I was sure it was just a contamination. And in red snow, you will undoubtedly always find many other green algae, also Chloromonas or Chlamydomonas species, but I'm sure in my field, I observed in fresh field samples also the vegetative stage of these red cysts. But until now, we don't actually know what it looks like. We always assumed it would look like this flagellated green cells, but we don't really know until maybe the Quarmby lab now has new information. But the red cysts, we don't have it as a live culture. Nobody on the earth. It's astonishing for such a phenomenon, but it's known from other algae as well that sometimes you have cysts. They have to undergo some kind of hibernation or stratification. You have to freeze the cysts or wash them with distilled water. I tried a lot of things, but it never worked out. But it's true, until now, the red cyst is just the red cyst.
Thomas, you say that some species, for example, grow better in snow places, but you show also some species that are found in snow places and grow better with higher temperatures. Then we can say that, for example, some this kind of place, the snow places, serves both as a place where the algae grow better and also as a bank of species.
Yeah, I mean, it's important if you want to study snow algae and you want to really study the cold adaptation. It's not good to look at alpine regions, for example, like the European Alps. We experience in the European Alps, those algae you sample there, they are not really psychophilic, they are cryotrophic. If you go to the polar region, and I will show you the difference between Arctica and Antarctica, also in Antarctica on the northwestern peninsula, we didn't really sample so many psychophilic isolates, but on Spitsbergen, because Spitsbergen is really polar, it's very far north. And in the high Andean mountain, I'm not sure how far you go up, but on Mount Pinatubo, I think it's in Ecuador, they sampled one Chloromonas art there. And I'm not sure because the isolates don't exist anymore. I don't know, maybe somewhere in South America, I don't know. But you are really, if you sample snow and you grow something, you really have to do this temperature check and see is it cryophilic or only cryotrophic. And we assume, because we wanted to understand at that time when I started, how long or when did the snow algae actually evolve in the evolution. And we assume that there was, I think 200 million years back, there was a glaciation of the whole Earth. And one could assume that during that time, this cryophily evolved, and then the algae, they draw back on these high polar and high mountain alpine regions where they have their refugium, where they survive basically the warmer times. And that's the only life area where they can, or the only space where they can live now. But many algae, of course, there's a theory of algae are everywhere. So algae are blown around with the air, and when they come down somewhere on an alpine region and they somehow find some nutrients and will multiply, but it's not their preferred area where they would grow usually. And it shows in the lab we can go all algae on high nutrient media. We don't need snow or aquadestillata to grow them. That doesn't work. We checked all that when I started because we thought it would be so complicated to grow them. But actually nobody had established cultures. There were two people in the 19th century, in the 70s, on Earth who had the cultures, and I was the one starting here, and the other colleagues are now retired. But interestingly, nobody grew them in the lab. Astonishing, yeah.
Yeah. Okay, thank you.
Shall we go on?
Yeah.
Yeah. So the next part will then be you see that overview again?
Yeah.
Okay, so the next field, I'll give you some field pictures, what it looks like where we sample our algae. So this is a view of the northern hemisphere of the Arctic Ocean, and I marked here this 80 degrees north. This is Spitsbergen, or Svalbard, one should say. It's an archipelago at the border of the 18th degree north. And that's also where we sampled. We sailed around the whole island here, Spitsbergen and Nord-Ostlandet. So it's high polar. Crimson Cliff also is somewhere here about. So it's really polar, in the polar regions. The pack ice is here. You see this whitish area, that's the pack ice. Not now, not these days anymore, I'm afraid, but we are really on the polar fringe there. 62 degrees north is much far south.
Thomas.
Yeah.
Sorry. There are a small window with some commands, a little of buttons in the up of the screen. Can you arrest there? You
don't see this
actually see, but we have just a bar of commands, a bar of buttons at the up of the screen. If you arrest that, because it's covering the title.
Yeah.
Sometimes.
I don't understand really what you mean.
Well, put again your presentation, please.
Yeah. So you see now the whole my presentation again.
Yeah. Can you see below your title? Our research starts in nature. Below that, a small bar.
Yeah, I saw that already. I don't know actually how to remove it. I try it. I can shift it.
Perfect.
Okay. I saw it before. I thought I was only me seeing it. No, sorry.
Don't worry. Don't.
Try to put on my laser pointer now again. Doesn't work. Okay, doesn't matter. So on the left, sorry, I'll be hitting a bit laser pointer. So on the left, you see what I just explained, the 80 degrees north here and the 62 degrees south here, north here. In Antarctica, that's very different. In Arctic, you have a polar ocean, and here on the Antarctic side, you have a continent, and it's surrounded by an ocean. So that's one big difference. And here on the South Shetland Island, where we sampled on King George Island, that is 62 degrees south. So it's much further north. South America comes up here. Then the polar region we sampled on the northern hemisphere. If you go in Antarctica at 80 degrees south, then you're far inland already on the Antarctic continent. Of course, the climate is much different. So that explains also why we sampled snow algae here on King George Island, which are not so cryophilic as the same algae which we sampled here. This is more maritime influenced and this is more polar influenced. So this is a difference between where you sample, not talking of sampling in the alpine regions like in Europe or maybe somewhere else or in America. Looking at Svalbard or Spitsbergen, Spitsbergen is only this one island here of the whole archipelago. As I said, Svalbard consists of Spitsbergen, Nordaustlandet, Barentsøya and Edgeøya, and also Jan Mayen, which is a bit further south. And it's ideal, it's great for geologists because the whole evolution of the Earth is there visible upside down for the geologists, but also for the biologists. You can sail around the island and go on land wherever, so that's the way we did it at least, and sample snow. And we actually sailed around the whole island to see what's different on the eastern coast and on the western coast. Most people doing research, they are in this research station in Ny-Ålesund, which is up here in the northwest, but they only stay in that area because they cannot travel further than with the zodiacs and around this fjord area here, which is a very shame. And I advise you, if you want to investigate snow algae, you have to look at different locations to understand how and where they occur under which conditions. If you look at the glaciation of Spitsbergen, there's a big difference between the western area, also influenced by the Gulf Stream going up here on the western coast and bringing some warm up there, as opposed to the eastern side. The eastern side is much more glaciated, it's very dry because all the western winds bring the rain to the west coast where it's raining down, and no rain reaches the east coast, as we experienced later on. So in the first year or the year before, we start preparing, of course, the expedition. We look at maps where to go. These black markings here are all snowfields with red snow. This is the northwestern area of Spitsbergen. And you see how often it occurs, this red snow. But we, of course, do this map study also with Google Maps to see where we can go, how easy accessible it is. We have to rent such a yacht with 4 people. We are on with a skipper, and it's a nice way to travel around. It's not expensive, as one might experience. And then we travel around this little island here in the north polar region. So in the first year, when we do expedition, we try to find good localities, and good is relative. Sometimes the weather is not good, but you have this extensive green snow here. You see a little bit. Sometimes you have to go with a zodiac on and see if there's red snow. This is lightly red here, slightly red. Sometimes you've got these little snowflakes, or snowfields, very small ones, and you have to climb up these fields here. Sometimes you also have to fly somewhere in the middle of the Spitsbergen. So we try to really visit as many localities to find as many different snow algae as we can to complete our collection with the highest diversity. And it is actually very diverse. Collecting samples can be easy and can be nice on such moss fields where it's nice and sunny and warm in summer. Sometimes you have the end of a moraine and a glacier here, can be very interesting and deep. You see it a bit later again. But you see already here, you have red snow down here, up here in the dry snow. Here, where it's a bit wetter, you have green snow in mass development, really thick green mass development of algae. And sometimes you see here the measure, we have to climb up the snow field, which is very steep. Also always watching out for polar bears and birds who can attack you. And sometimes you try to find the same or try to point at some certain samples, but people don't really see what you want to show them sometimes. But yeah, so we collect samples, but we always have to really avoid hazards. And in polar environment, it's really strong. This is our skipper. He was always climbing up the sail mast really to watch us when we were going on land with the zodiac. And then he called us sometimes. And in this case, he called us saying, hey, there are polar bears around. This is a photo montage really. This is on land. I'm on the boat already, so I'm not in the vicinity of the polar bear. But we were surrounded suddenly by 4 or 5 polar bears, not attacking us, but they were behind us. So then we had to go on the Zodiac and on our ship to stay away from the polar bears. Also, the sea is very hard to maneuver, so we were lucky to have our experienced captain really to maneuver us around the icebergs safely. Sometimes the environment is horrible. You get stuck in mud and you fall over. Obviously, always colleagues have the camera right to make a photograph of these funny pictures. Me dropping into water, my boss at that time being stuck in the mud. Luckily, we got out of it because it's really dangerous to be sucked in, like you're sucked away in this flowing silt sand. It's really horrible. And sometimes also the land animals are dangerous, like these walruses, or especially these skuas. They attack you constantly, and you have to chase them away. That's really a nuisance. But of course, you're in a fabulous environment, and these pictures, yeah. Nice, cute polar bears, but they are very fast on rocks, on land, and also in the sea. They are fast, and they look cute, but they are really the most dangerous animal on land on our Earth, on the polar regions, and they undoubtedly will hunt man and eat them. That occurs annually, every year in Spitsbergen. The people get attacked by polar bears because they are not armed and they don't shoot at the polar bear. But that's another story. Red snow, that is a phenomenon when you actually find red snow. That is that's really the mass phenomenon. It's very striking if you see it, because you think somebody spilled blood all over the glacier, and you can assume that here you have a concentration of some 10,000 or nearly 100,000 cells of these red cysts per milliliter. It's a mass development, and it's really hardly to understand where the algae draw the nutrients from to have such a mass development. This was especially in the middle of the nowhere, on a glacier on the top, far away from bird colonies, far away from any influence of nutrients, and you measure the snow and there's no nutrients in there. So it's a really big question how they can, what kind of nutrients they really use. And we assume they might use mineral dust, so mineral nutrients from the surrounding rock. That's the only explanation we have at the moment. This is a bit different. This is also in a mountain slope, also hardly no vegetation around, and you have this red and orange snow. You see this typical coloring. You see also here in this area it's dark red, and in other parts it's orange. Why is it not other way around? Is it a topology and the wetness of the snow? We don't know really. If you microscope them, you find the red cysts and you find this orange cysts, and these are exactly the two species of the new erected genus Sanguina, Sanguina nivaloides, which is most probably more or less synonymous with Chlamydomonas nivalis, at least pro parte. And then the orange cysts are the Sanguina aurantia meaning orange. So these are the two dominant species forming the red-orange snow. As I said before, we don't have live cultures of these cysts, so you can't order them from our collection, only the cyst material for very special occasions. Going to the northwest of Spitsbergen, there we found on one of our cruises the Makarov Brain, the Makarov Glacier, and here is a big bird cliff, this big cliff in the background. This is full of birds, and of course a lot of nutrients come down with the rain from the cliff and is being spilled onto the glacier. And this is understandable that snow algae can develop nicely here on this glacier because it's being flooded in spring and in summer with nutrients, but not in winter anymore. Just to give you an idea how big that is, this is about a kilometer long, and the snowfield here is about 200, 300, 400 meters wide. And we calculated it, and the annual productivity of these mass developments of algae is about 100 kilograms of fresh mass per year. That's what we produce here in our pilot plant for industrial cooperation as well. It's fairly reproductive and productive in a way. And you see it here on the bottom at the end of the moraine, where the melting of the glacier comes down. You have the meltwater, and you have this green snow algae develop here, clinging to the water, to the snow, and to the ice. And this is where also the strain 50 grows as well. We find it very often in Spitzbergen. On the snow, you find Chloromonas and Microglena and Rhaphidonemma in the green snow. And in this brownish here, this is not all dust from the rocks, but also Ancylonemma species. There are 2 species described in this genus now. Formerly Mesotinium bergrenii, it also has been transformed to Ancylonemma, and they make this violet snow. That you see it, it's a little bit violet tint, and this is typical for locations where Ancylonemma occurs in really frozen ice, not in snow, but really frozen ice. The focus on this end, and you see how dense the Chlamydomonas and Chloromonas and other flagellated green cells here really grow in this ice slush at 0 degrees. It's not warmer than 0 degrees, and it's a perfect temperature, obviously, for them to multiply. Another location in Hornsund, this is on the southern area of Spitzbergen in Gnallodden. There was a very windy and wet and rainy day, and we observed this flat snow field. I actually want to fly with a helicopter. I'll show you a bit later a picture of that. And from when you're on the ground, you don't see very much. You see this white snow field and this kind of melted areas, which are a little greenish, but you don't see very much when you're on the bottom. But you can sample, of course, green snow from these areas and from the red snow, and you find these red cysts and orange cysts and also other algae in between. But if you use a helicopter, a so-called drone with a camera system, and you make aerial photos from the top, you get a complete different picture. And this was very interesting for us to do that one year. This picture, this big panorama picture here, consists of more than 500 single pictures being all put together. And you see this here is our zodiac. This is about, the scale is 10 meters. So it's a quite huge snow field with a different topology. You have these melted areas here. This is a digital manipulation of the picture where we drew the red and green color channels. We intensified them, and you see that the green multiplying cells are here in this wet melted areas of the glacier. In the dry areas, you have the reddish red cysts. They, in normal view, appear like this red tint, but you can increase the color here in the photo program. So you see a high diversity in the topology and in the whole type of the snowfield if you look from the top. This is really interesting to do that, and you can, of course, use these 3D pictures to use them in models and in GIS applications. We didn't pursue that anymore, unluckily, but it was worth it to view a snowfield from the top as well. A very, very different picture now when we go from the west coast to the east coast. I told you the east coast is rather very dry, and we had a geologist here, my colleague from the Geo Center in Potsdam. He's a geochemist, a paleogeochemist, and he told us a lot about the geology of Svalbard, and he said the whole eastern part is really arid and cold desert. You have hardly any rain coming down, so the whole glaciers are very small. You see in the countryside you have some snow rests, but everything is evaporating very fast. The air is very dry, and we also hardly found any red snow and snow algae on the snowfields. It's very dry, and obviously the dryness and also the rain coming down in summer plays a crucial role for stable snow culture, snow algae establishing populations on the glaciers. That was in the western and northwestern part of Spitzbergen. You observe it very often. Along the eastern coast, we hardly observed intensely red or green snowfields. It's a very different condition. Hopping back to our years coming, developing over from our field to lab, we are still in 1 year, so we sample a lot of samples, red snow, green snow, and soil samples, bring them back on the cruise, on the vessel. There we preparate the samples, we preserve them in glutaraldehyde, for example, for our archive. We take samples for molecular analysis at home. We freeze them on board. Or we isolate single cells with a microscope. We make photos. We do chemical analysis. So it's the whole day, like 8 hours in the snow field, and then the evening until midnight, because it's still light, the sun is still out. Until midnight, we do all the work on the ship. So it's a long day, but I don't complain really doing this polar expedition. It was a reward enough for all the long working days I spent there. And then when we come back from the lab, then we try to isolate the cells, of course, making them clonal and exenic. Exenic means free from bacteria and fungi and other algae, and takes about 2 years to get from a field sample, which looks like a pizza pie here, with all the different algae and fungi growing on it. And then if you isolate single cells, then you end up after 2 years with a culture, and then we try to find out the correct culture medium, and after 2 or 3 years, they end up in our collection, where they get a strain number, and then you can order them. We also culture some mosses for those people being interested in polar mosses. Also some cyanobacteria and fungi. And recently, I'm having just now a student who's isolating the bacteria and fungi from such samples to also offer this coexisting organisms, to understand maybe about nutrient fluxes between fungi and yeasts and bacteria to the algae and vice versa, to understand where the nutrients come from. So from these locations, we have then pure cultures, like here from these locations in Stuphallet. These are different locations because here you have calcareous deposits coming up. So this is very alkaline, and compared to the rest of many locations where it's more shale and sandstone, which react more acidic. This is more alkalic. And in these more alkaline locations, you have Raphidianema very often, and Stichococcus. Raphidianema was also a genus which was very hard to understand the species and the phylogeny. And this year we revised this genus of Raphidianema and came up with a suggestion of 5 Raphidianema species, all based on molecular analysis, together with the colleagues from Vancouver. Also, it just was released in a print version of the Journal of Ecology now this month. It's a free available version as well, and you're also welcome to download it from Journal of Ecology. It's quite nice. On these permafrost soils surrounding these locations, you can isolate also these permafrost algae, many cockle algae, which are typically green when they multiply, and when they form resting stages due to desiccation, when they fall dry, or when the nutrients deplete, then they form this carotenoid-rich cysts or spores, where they accumulate lipids and also accumulate a lot of secondary carotenoids in these lipid globules, becoming red. If they become nutrients available again, then they become green again. So they change in between green and red very easily. Taking nutrients out makes them red, putting nutrients in makes them green, putting more light on them makes them red, taking the light away makes them green. Again, this is not true for the snow, red snow. This is a typical different system, but for the soil algae, this is very easy. Going to Antarctica, I told you about King George Island. This was a very interesting location right on the sea level. This is a very sunny and very calm day, but here on the west you see the ocean, the polar ocean, and a glacier here from the inland flowing into the ocean. Not a final moraine like in Spitsbergen, but the glacier flows right into the sea. And of course you have sea spray going on the fresh water from the glacier. But even in this very highly osmotic stress system, where you have constantly marine water with 35 promille chlorine or chloride water coming onto the fresh water. Actually here also Chloromonas or Chlorominima, I have to say, colleagues from Chile, I think the colleagues are, they published a paper now, and they described a species of a Chloromonas-like algae from this area, and we had isolates and we compared that, and most probably it's this Chlorominima, as they call it, because the Chloromonas-like cells are very small. So they made up this new genus. Very remarkably that this alga can grow easily in this highly osmotic stressor system. Yeah, and then in the third year, basically, the algae end up in my fridges. They grow in these agar slants as unialgal. So it's only one species in the agar slants, and most of them are exenic, free from bacteria and fungi. Some cultures I have from the same species also with the bacteria and fungi and yeast. That's where we isolate these coexisting organisms now from. But if you order them, you can choose between exenic or non-exenic strains as whatever you want to investigate. They are basically cultured in these fridges around 2 degrees plus, but not higher than 4 degrees. Sometimes in cool rooms like this for larger growth masses if you want to culture them. And they're also being cryopreserved in liquid nitrogen if necessary and if possible. So some news about the algae collection. The CC Cryo, that's the acronym of the culture collection. If you just Google CC Cryo, then you will end up on our collection, or you Google cccryo.fraunhofer.de, then you will come to the online database. That covers about 424 strains from 177 species in 100 genera, not all being snow algae. This diagram here shows that the majority still belongs to the classes of Chlorophyceae and Tribuxyophyceae. That's the two dominating classes, basically representing the snow and permafrost, the cryophilic and the cryotrophic algae. As here in this diagram from the culture collection, about half of the strains, like more than 250 strains, are really psychotolerant only, but 40% are psychophilic. These are the Chloromonas and Chlamydomonas species being in this group here. But many other organisms are also mesophilic ones, and also some thermophilic algae and cyanobacteria we culture, and also the mosses. But we have a clear focus really on the cryophilic algae. And I think I can say that the collection is really unique worldwide in its extent and diversity. And since we opened it to the public in basically like 10 years ago, we serve the whole community from around the world, from any continent with strains for lab studies. We mainly culture at 2 degrees, as I said, but we have also culture facilities at 15 degrees or room temperature, or for the thermophilic ones at 32 degrees. The strains are suitable for basic research, but also for applied research. If you want to look for special components or ingredients you want to sell. We do have hold authentic strains. These are living cultures of type strains. So when you describe a new species, like these colleagues from Chile, then they can deposit the live strain in our collection. That's not the type strain, because the type strain, it has to be in a physiologically inactive condition. So obviously it can be dried as a desiccation material and given to a museum in your country, or can be also cryopreserved in a collection. But usually we only receive the authentic strains, so that people can order the living strain to compare their phylogenetic studies, for example, with the type strain. We do not hold patent strains because we don't have the facilities to really make patent strains really constantly available. But there are other collections like the CCAMP in the UK and Scotland at Open, for example. That's where we put our patent strains. But as I said, if you describe new snow algae and you want to deposit an authentic strain, then we are good for that because we have the good culture conditions for that, and we make the strains available to the other colleagues as well. The online catalog, as I said, you can access it by there and search conveniently for any type of word you type in the search field. If you search for Antarctic or Arctic or snow or glacier or any genus, and you just type it in the field, and then you get a list of species that are available from our collection. We are a member of the World Federation of Culture Collections and also of the European Culture Collection Organization. As I said, strains are available for the scientific community, but also for industry, for schools, art projects, whoever wants to use these strains for any purpose. And the question why we do snow algae research at Fraunhofer, it has historic reasons, because my boss from our institute was my PhD advisor at the Humboldt University, where I did my PhD on snow algae. And he got a call to become a director of a Fraunhofer Institute. And then he took me and my collection with him to this Fraunhofer Institute. And that's where we erected this collection and have it now. Fraunhofer usually is doing much more applied research, very technical. As said down here in Germany, Fraunhofer next to the Max Planck Institutes, you know, Max Planck Society possibly. Max Planck is doing worldwide basic research. Fraunhofer is doing applied research. That's in Germany, the difference between Max Planck doing basic research and Fraunhofer Society doing applied research. Fraunhofer is very technical usually, but many institutes also do life science studies now. I'm very exotic, no way, but I have also the urge to do applied research, because our job at Fraunhofer in Germany is, our job is to develop products for the small and medium enterprises, the companies in our industrial landscape. Where do our strains come from? Obviously from polar regions, so many strains come from Spitsbergen and Svalbard up here. Many strains come down from Antarctica, where I did an expedition in 2006. And then some colleagues sent me strains from the Rocky Mountain areas. Obviously also not so psychophilic, I must say. They are more cryotrophic rather than cryophilic. From the Andean Mountains, we have one strain, which is not a real snow alga, really. From the Appalachian Mountains, it's a low mountain area in the eastern part of the United States of America, not really snow alga area. Also the alpine areas in the European Alps in Germany, as I said, not really psychophilic, but you find Ancylonema here, this violet dot, and a lot of other Chloromonas species, but they are all not very cryophilic, they are rather cryotrophic. And some of them, these are more thermotolerant or thermophilic species, also from Australia and from Java, from Indonesia, and so on. Also from New Zealand, from the New Zealand Alps, we have got some Rhaphodinema isolates, and as I said, also around from Antarctica here. So quite diverse. We unfortunately hardly have any strains from South America. That would be really nice to have some isolates from there. But I know that some colleagues in Chile, and I think some are also listening maybe, but are working on snow algae. So we've got some colleagues there as well. And unfortunately, nothing from Asia as well. It's hard to get strains from China, of course, from the Himalaya. That would be quite interesting. But some people in Japan are working on snow algae, in Korea as well. So snow algae research is being performed in Canada, in Europe, in South America, in the United States, even in Middle, on the West and East, and on Japan, all over Europe, and New Zealand a little bit. So it's a small community of snow algae researchers, but everywhere. Our database, the external database is very small. The internal database is depicted here from a FileMaker database. It's a relational database with a lot of tables giving information on taxonomy, on the metabolites of each strain, on where they had been found, who collected it, also on the Nagoya Protocol requirements, on collecting permits. As I said, what metabolites have been found in the algae, what are the metabolites good for, and so on, and so on, and so on. So it's a huge internal database with a lot of information we have. Some of the tables I have then on my screen is this main database with picture of the alga, of the location, and then lists of the metabolites, for example, a log file of what I did to the culture, link outs to other databases like NCBI. I had to have a link out because I have to look up the genetic data as well. They are incorporated in our database here as the genetic data, which are uploaded as NCBI or EMBL at the databases for genetic resources. We have a quality control. The strains are being checked on contaminations at least every second year. Metabolites, as I said, on cryopreservation. So it's a lot of information we gained around the snow algae. And if people have questions regarding snow algae in our collections and our strains, then feel free to contact me. I think I can give you a lot of information if you have special questions. So what do we do, or what did we do in the last 20 years? As I said, we work from adaptation strategies to biotechnological applications, and the basis is always the strains themselves, the expeditions, and then we have to do taxonomy and phylogeny to know what we're dealing with. We cryopreserve the strains and have them in the collection. My technical assistant, Karina Teufelhardt, she's helping me with the collection. Then we have on the other side, some very exotic studies with a German aerospace research company in Germany, the DLR, comparative to the NASA, for example, in the United States. And we sent some of our strains on the International Space Station and pinned them on the outside of the station to see if they would survive there. That was very exotic project. There's some European projects on fatty acid. Other industry projects on the mass production for cosmetics mainly, for cosmetics products. We develop suitable photobioreactors, these PBR photobioreactors, to produce the algae, and also develop pigments and antioxidants for aquaculture, for example. That's for the industrial part. A lot of basics also address transcript studies and proteome studies, and also to access basically the cold active enzymes and the genetic data for that. I'm doing this with my colleague here, Dana Wenzel. She's also a technical assistant. She helped me with these studies and also with the industrial production together with Felix Jord, who was my engineer. And he's doing the mass production for the industry. And currently I'm focusing more on the cold active enzymes and proteins, which are ice structuring proteins, also known from other algae. I'm coming to that later on in my talk. But currently we also have a lot of transcriptome and genome sequencing and data mining on this data to find special enzymes we can use in industrial applications, and also other proteins which are related to stresses. And I do most of this currently with students who are doing the bachelor and master thesis in my work group. So there's a long way, and that's basically, I think, the end of my first part of the talk. After 3 years, you are from field samples, you have pure cultures in the lab, and we do this pre-characterization. Of course, we look what is the name of the culture we have. We do some first fatty acid analysis, some first pigment analysis to see if there's anything special in the strains we have. We look which medium of culture medium do they grow. Are they really cryophilic or are they just cryotrophic? So to know what we're talking about, and if we observe anything strange like these EPS, this gelatinous matter surrounding the cells, a lot of them make a lot of this gel-like EPS, this extracellular polymeric substances. Some people call it extracellular polymeric sugar. Some people call it, but you know what I'm talking of, this gelatinous slime, whatever surrounds the algae. And then when we know what we're talking about, then we go on with the research and go into the application part. So I'm over with the first part of my talk. I would switch back now to look if we find some
15 minutes.
And then we can have a break. So
okay. No, we until now we have not any questions, but maybe after the break, okay?
Yeah. Anyway, yeah.
Ok, pessoal, então 15 minutinhos de intervalo para a gente descansar, ok? Tomar um cafezinho, já já a gente volta. To drink a coffee also, I was talking about that.
Yeah. So I think 15:30 would be fine. I think we're getting well through the talk, and I'm happy to answer the questions afterwards, after the break. So see you in 15 minutes.
Okay. See you. Olá, pessoal, estamos retornando aqui novamente. Vou trazer o Thomas para cá de novo. Welcome again, Thomas.
Hey.
Let's go on.
Any more questions, Rose?
No, not now.
Okay, I posted on the chat the two citations of the two publications I mentioned for people who are interested in downloading the publications.
Yeah, I saw that, and I will publish it here. Okay?
Okay, so we should go on.
Yep.
So you see that?
Yep, okay.
Okay, I hope you could follow my talk so far, and as we said, please feel free, but I think we have at the end we have a lot of time left, so feel free to ask any silly questions you have, whatever you want to know. I'm happy to answer them. So in the first two parts, I told you what the snow algae actually are, where they came from, how they end up in our lab, and so we have the cultures now in the lab, and of course, as I said, at Fraunhofer, we are looking for products. So anything we can transfer into the industrial area, have a mass production of algae and the metabolites to use them for industry. I'm going into that at the very end of the talk, a bit more into it, but in the next part, I will give you example for metabolites from snow and permafrost algae and also a little bit of thermophilic cyanobacteria. We're talking here of pigments, of course, of antioxidants, if we're talking of the carotenoids. we're talking of the omega-3 fatty acids, or know that, or the polyunsaturated PUFAs, the polyunsaturated fatty acids, which are important for our human diet, like the EPA, the eicosapentaenoic acid, or the other 3 omega acids, the DHA, for example. The extracellular polymers I told you about, eye-structuring proteins, I will tell you, a special thing that we find in the snow algae, only in very psychophilic ones, a very special type of proteins. And then I'll also give you some information on the genomic and transcriptomics we do at the moment. So the snow algae, we know they are special because they are extremophiles. They adapted to the special stresses they have in the environment. As I said, the growth temperature for the optimum is generally in the range of 0 to 15 degrees. And the maximum depends on the species and the isolate. They can sometimes grow a little bit above 15 degrees. Some die at 10 degrees already. Some strains die at 20 degrees or 23 degrees, but they all have their optimum below 15 degrees. That makes them psychophils. The nutrient and light stress, I said, they've got high light stress on the glaciers. You can imagine that a lot of light is going onto the glacier. And of course, the algae react in a way that they make carotenoids, the orange and red colored pigments, and the secondary pigments. Some of them act as antioxidants. Nutrient stress sometimes also induces fatty acids being produced, one more than the other. But also in the enzymes, you have photolysis, where the algae oppose the light stress, and they can make radicals to inactivate the radicals that are being produced by high light stress, and they have enzymes who diminish these. And we also have photoreceptors. It's a very new field. We have blue light and red light photoreceptors, which induce certain physiological processes in the cells. The low temperature, as I said, the temperature is a decisive stress. It's a cold stress. And due to the freezing of the water, you have high osmotic stress because the nutrients accumulate and increase. So you have osmotic stress, and on the other side, you have desiccation stress as well. Cold stress or freezing stress always is also desiccation stress. You might know it if you're in the Alpine region going for skiing, then your skin gets very dry and you have to apply cream on it. So you've got all this dry air around. So these are the stresses due to low temperatures. And the low temperatures, they induce the production of different cold-induced metabolites. Again, also the lipids in certain conditions. And of course, all the cold-active enzymes, among them also the ice-structuring proteins. And other genes, of course, for all different other enzymes which are activated during cold stress. Coming to the secondary carotenoids, a very obvious thing, the red snow has accumulated a lot of astaxanthin. That's a carotenoid, a secondary carotenoid, a so-called ketocarotenoid. The carotenoids have a double bonds in their molecular structure, and these double bonds make them good radical scavengers. So all the carotenoids, also the beta-carotene you find in carrots, for example, they can fight radicals being built up in your body. So in that way, the algae producing these carotenoids, you have here a culture of a snow, or no, it's a soil algae actually, when they have a lot of nutrients and under low light conditions, they multiply and form these green cells where they do photosynthesis and multiply. Once you take the nitrate and the nutrients out and give more light, then you see little fat globules, little lipid globules being accumulated, containing very often oleic acid. And in these lipid globules, all the carotenoids are stored because the carotenoids are all fat soluble and they are accumulated in these fat globules. And this makes also up what you see as red snow. These are the cysts with a lot of globules, fat globules in them, and with a lot of astaxanthin and other secondary carotenoids being stored. Some species don't become green, but they still produce beta-carotene, for example, and also lutein, and often also alpha-tocopherol, vitamin E, and all these act as radical scavengers. In green species, which don't become red as here in the soil alga, but the erubescent species, erubescent means becoming red, and they produce the secondary carotenoids like astaxanthin or canthaxanthin, and also other xanthophyll cycle pigments, which make them red. For applications, these pigments are interesting because they have high antioxidant activity. They are good for the development of zooplankton. You need zooplankton if you want to feed salmonid fish, for example, in aquaculture. You need to raise young fish, you need zooplankton, and the zooplankton, of course, feeds on phytoplankton. So the phytoplankton, our algae, are the starting point for all the fatty acids and the carotenoids. The zooplankton accumulates these fatty acids and the carotenoids from the algae and become reddish. And the fish larvae and the adult fish, like salmon, they feed on the zooplankton and take up the red carotenoids and the carotenoids and become red, and also take up the fatty acids which make the salmonids fish so important for the human diet. But the PUFAs, which you eat from the fish, actually come from the algae they eat. And of course, for selling fish like salmons, the appealing pigmentation, the red salmon flesh, is due to the astaxanthin. In aquaculture, as I said before, the phytoplankton is the basis. They transform CO2 and from light energy into proteins and the pigments and the fatty acids. They are the basis for the zooplankton, and this is being eaten by fish egg or by other crustacean shellfish which are being cultured. And also the shellfish, which are often sold now for the humans, like shrimps and prawns. They have this nice reddish color from the carotenoids, and also the salmon, as I said, also coming from the red pigmented algae. These four ladies, I didn't photograph them myself, but they are anonymized here. You see with the mud they scrubbed in their face, and they sit in a pool where also cyanobacteria grow. This is the desert near Las Vegas in the United States of America. And I was there on holiday, and I sampled from one of these hot pools. This is a thermo pool, 30 degrees. And in there grows these nice cyanobacteria, also being extremophiles, not snow algae, does nothing to do with the snow algae, but we culture extremophilic organisms as well, and among them are these cyanobacteria, and they have these bluish pigments, and depending on the species and on the buffer system or the extraction system, they are all water soluble. And then you have, depending on the polarity of the extraction solvent, you have violet or pinkish or greenish pigments or bluish, and also these pigments can be used in the industry, for example, in lemonade or in beer. I don't know if you have that in the American market, but in Europe you can buy blue beer. It's all about selling points, having new trends setting up, and of course in the trend disco community, blue beer is interesting. So a company selling blue beer has an advantage. Instead of selling just normal beer, they want to have new products, and these are trends we can serve. The advantage of these natural pigments is that they're non-mineral based, because usually, especially for cosmetics, you have minerals being put in the cosmetics, which is not really good for your skin, basically. And also the mining of the minerals is highly unenvironmentally good. It's not good for the environment. So this would be a good alternative, produce natural pigments. Going to the fatty acids, you can do a GC analysis of the fatty acids, of the profile of an alga. This here is a psychophilic alga, also a Rhaphidonemma type, very unusual, that we just newly described. It grows on permafrost soil and also on snow. And if you grew it under certain conditions, and if you take the nitrogen out, here the blue bars are with nitrogen or nitrate in the medium, and the white bars are without nitrogen. Normally, the Credo was, if you take the nitrate or the nutrients out, then you have an increase of the more unsaturated fatty acids. This is not really true for the snow algae we found out, but you have a typical pattern of algae with linolenic acid very often being a majority of the fatty acids, but also EPA, as I said before, eicosapentaenoic acid, and DHA, which is not produced by this alga, but EPA is very good for your human health and is being sold in the pharmacy as little capsuled in fat. Usually these are used or gained from fish scrap, and fish they bycatch they get when they normally go fishing, and the fish are being pressed out, and the EPA and the DHA from these fishes and from the fish scrap and bycatch from little fish they can't really market, they are being put into this capsule you can buy in the pharmacy. Arachidonic acid, ARA, is also important for the infant development of the brain, for babies. Baby nutrients and baby milk are often enriched with arachidonic acid for healthy development of the nervous system and the brain development. It's not so good for human adults, because it's an antilog to EPA, so you can eat a lot of arachidonic acid, for example, avocado. It's rich in ARA, but it's not good to eat too much ARA. If you eat enough EPA, it's okay, but you have to be careful with your diet to eat too much of either of these fatty acids. But you can, in general, you can use algae for the production of these important fatty acids. Some of the algae produce very long-chain fatty acids. This is very unusual. For example, the C24, we have to confirm that if it's really nervonic acid. Nervonic acid is a fatty acid which is also in adults important for the healthy development of the nerve cells in the brain. Has something to do also with multiple sclerosis. If you have a lack of these important fatty acids, then the multiple sclerosis might develop, but that's something we don't follow up at the moment. Pharmaceutical research is very complicated. But it's nice to have algae from a natural system which produce these very important fatty acids. I said before, the Credo, normally if you ask people in algae, they say, yeah, just take out the nutrients and stress the algae, then you get unsaturated fatty acids, because the more double bonds you have, the more flexible the fatty acids are, and also the membranes. A lot of the polyunsaturated fatty acids are located in the membranes of the cells, and according to the membrane fluid mosaic fluid system you might have heard of, one says the more fatty acids with double bonds you have in the membranes, the more flexible a membrane stays, even at low temperatures. So Credo was low temperature adapted algae have a high amount in unsaturated fatty acids, and at warmer temperatures usually there are less double bonded fatty acids. But this is not really true. We found from left to right, here on the left you have psychophilic species like Chloromonas nivalis, it's called here. Actually, it has a new name now, Chloromonas remiazii, but it's cold adapted, it's psychophilic. And you see, even under full nutrient conditions, you have a high amount of this orange part, which is saturated fatty acids. And if you take the nitrate out, you have a higher proportion of unsaturated fatty acids. So that's a typical system, you know. But in other algae, it's not necessarily the way. Here in Rhaphidomnema, it's also psychophilic. This filamentous algae I showed you before, different species of the genus, they have, even under high nutrient conditions, they have a biggest proportion of polyunsaturated fatty acids. And if you take the nitrate out, you get a less amount of unsaturated fatty acids. So the difference is not so big as you assume, as you expect. normal algae. In other algae, which are less cold adapted, like Xerocystis, this is a soil alga here, you have a high proportion of saturated fatty acids, which is increasing even under nitrate deplete conditions. So it's vice versa, what people normally know or expect. And also Macrochlorus multinucleata, it was called at that time when I made these diagrams, I didn't change the names, I see now. This one is called Macrochlorus rubriolosa, which means reddish, and it's a mesophilic alga found on snow, also making big dark red cysts. And in this species, hardly any difference between nitrate deplete and high nitrate full conditions. So you have to be really careful what alga you're working with, and actually I think it's all species specific, and especially in extremophiles you have to be careful to apply the normal thinking of an alga biologist. Rather look into your species and see what funny things these extremophiles are doing. Sometimes they do really unusual things. I told you of these extracellular polymers, this EPS, this gelatinous matter. I forgot to put in a microscopic picture, a close-up at least here in this picture down here. You see a microscopic picture, and the greenish parts here, these are single cells of algae, and they are covered by this gelatinous matter, really. Even single cells have a thick layer, several times as thick as the cell itself, protecting the cell on the outside with a gelatinous matter. Of course, it's protecting the cell from desiccation because you have this jelly around the cell. It's also a mechanical protecting, because often the soil algae land up on the bare soil in summer in the moss fields, and they have mechanic stress there, desiccation stress, but with this cover of gelatinous matter, they are well protected from desiccation and mechanical damage. And this picture is here, what you see, this blobs here is air trapped in this jelly matter after centrifugation. It's very funny, if you grow the algae and you centrifuge the algae off, then you have masses of this jelly being left over, and that's consistent of different sugars and sugar alcohols and sugar acids being connected with the sugars, dominating here by galactose, and other algae. Trehalose also accumulates within the cells, but in this gelatinous matter, other sugars are dominating, and also the sugar acids, uronic acids, for example, it's often connected to the sugars to form this matter. And in this particular strains I studied there, if you centrifuge it on the top of the centrifuge tube, if you take it out of the centrifuge, you have this kind of sponge really. You can take it off with your hands, and if you microscope it, you see it's very firm in structure. It's very, you can grab it really, and you can maybe use it in industrial products as well. For example, in foams or mousses, you apply your skin or your hair, and instead of putting microplastic and other petroleum-based jellies in there, you can apply these natural ones. This is not being realized because the production of these jellies is too expensive in comparison to petroleum-based traditional jelly and jelly makers. A problem now is microplastic. You all know that our sea is full of microplastic that also comes from cosmetics in large parts. All, when you buy cosmetics and it's all kind of jelly form, it's often full of microplastic, which ends up in the sea later on. And we postulate, for example, you can put these jellies from these natural algae also in cosmetics as a water binding component, naturally as a structural stabilizer, so that your cream becomes jelly-like, or your hair jelly becomes stable, and we could maybe replace microplastic in future. But again, it's a question of money all the time, and industrial products, the point which is decisive if a product enters the market later on is the money. And currently we still have too much fossil oil and petroleum-based basics available on the market from an environment. So there's no real urge to replace it completely by natural sources. I come back to this problem a bit later. I'm coming now to a question we had at the very beginning. We asked ourselves, of course, in the natural habitat, the algae are exposed to this constant periodic freeze-thaw cycle. They live in this snow, which is really hard to survive in. I mean, if I see myself sitting on a glacier in Spitsbergen, I have no food with myself, and I'm sitting there. I could drink the water from the snow, of course, but basically after half an hour in this cold environment, having no resources around me, I would die. And these algae have managed to evolve to an organism to survive there very well at 0 degrees, living basically on no nutrients, having a lot of light available and water to a certain amount. But how can they actually survive there, being freezing and thawing all the time? And we asked this, and it's hard to answer that question really. What they need for not being damaged when freezing, because that can occur during the day if the wind changes to northern winds and bringing polar snowstorms in, then suddenly all this melted water freezes, and of course the algae are freezing as well. And what makes them survive these conditions? And we know that they can lower the freezing point of the cytoplasm by sugars. Trehalose is often accumulated as a structural stabilizer as well, and also a lot of other sugars like sugar alcohols like glycerol. You know that from as an antifreeze protectant in your wash wiper water on your car. Also proline, some amino acids accumulated, and all they protect the algal cytoplasm to freeze during these small freeze-thaw cycles. We don't know what else is there, but what we observed, and other people also observed in other organisms, are so-called ice structuring proteins, or antifreeze proteins they were called, AFPs. As said, EPSs, these gelatin matters, is also made to protect them from the outside, but we really don't know what else is the algae invented over the years, a million years of evolution, to make them survive these harsh conditions. These AFPs, as I said, the antifreeze proteins, that was the original name when they first were detected in Antarctic flounders, this winter flounder, which is living around the Antarctic waters, and people asked themselves, how could the fish survive freezing temperatures in the Antarctic Ocean, which is -2 degrees, -1, -3 degrees in some parts. And people in the 1970s, they found proteins in the blood of fish, and they found that these proteins prevent the blood from freezing. They depress the freezing point of the blood, and also in some insect, in the hemolymph, to lower temperatures. So the blood really freezes not at -3 degrees, but only at -5 degrees or -8 degrees. That prevents the insects, like also here in New Zealand, it's a giant wheater, the tree wheater living in the alpine regions of the New Zealand Alps, and it can freeze completely. And also there they found those antifreeze proteins highly hyperactive. They depress the freezing point to a strong degree and prevents the animals from freezing. In plants, one then looked and one found also in winter rye and ryegrass, in the apoplastic system, not in the cells themselves, but in the interplastic space between the cells, they could find proteins which have the same effect. They depress the freezing point to a certain degree, not as much as in the animals, but also by 2 or 1 degrees. But also they influence the formation and the growth of the ice crystal. That was so interesting to see. And I'll show you in a moment what happens really. They actually directly influence the growth of the ice crystals and make them not so damaging. So you find these antifreeze proteins or ice structuring proteins, which are they termed, if they have a slightly different effect, or ice binding proteins, again, some bacteria, the yellow ovals here, they produce IBPs, and obviously these ice binding proteins enable green algae, which are diatoms living on the bottom of sea ice, and they can connect via these ice binding proteins to the ice crystals. So they can connect them, they don't sink down, they stay up near the ice flow on the sea surface, they are near the light which comes through the ice flow, and so they are connected to the ice crystal, to the ice flow on the Arctic ice or Antarctic ice, and they can perform photosynthesis and also serve as nutrients for other grazing little animals living on the bottom of the sea ice. You see here a microscopic picture as well. You see the diatoms in the pack ice on the bottom, and the pack ice appears this brownish green here. This is all this algae being on the bottom, this little ice flow turned around basically, and they live on the bottom of the ice flow. And also coming back to our snow algae, in this psychophilic algae, again, our strain 05099 of Chlamydomonas clinobasis, or Microglina, it's now called in the new genus. They produce these ice structuring proteins, because what happens with these proteins, if you look at it, I will show you in the next slide. We always wondered, why are these algae actually producing them? Because the ice structuring proteins doesn't really protect them from the freezing stress. We don't believe that. It's more what we believe is that also the freshwater algae, the snow algae, they can connect to the ice crystal, so they stay in the vicinity of an ice crystal all the time. And due to physical laws, if you are connected to an ice crystal, and even if you're frozen in ice in the cell closed area, you will always have 1 or 2 monolayers of liquid water. And the algae, of course, the algae need liquid water for their metabolism, not the freezing water. They can't use frozen water, but need the liquid water. But if they're in the vicinity and if they're linked to an ice crystal and stay there, they are sure they have always their water with them, no matter where they are drifting or where they are blown by the wind. And what happens by the end of the summer, when the glacier melts and the snow algae are in danger of falling on the bare ground and being dead, they can be blown with strong winter or autumn storms, because in autumn, northwest of Spitsbergen, from the west, really, the western storms from the Atlantic come in and take up the snow crystals and disperse them all over the mountain ranges in Spitsbergen and take them, the algae, to higher ranges in the mountains, where they can overwinter, being the snow comes in winter and it covers them, and then they can survive winter. So we think actually the ice structuring proteins also act as binding proteins. These snow algae connect to the ice crystals and are safe because they get water and are also dispersed around in the area. Because you always ask yourself, if an alga is being on a glacier, is being washed down, how does it come up to the mountain areas again? It can't fly up where all the snow fields can be observed, because we observe red snow and green snow also very high up in the mountains, where the only possibility is that the algae are dispersed with winds in the high mountain areas. And where it's good conditions later on in spring, then you get the green snow again, or the red snow. So how do these ice structuring proteins work? These are diagrams from 2 publications here, from Gia and from Wathan, from 2002 and 2003, and they depict it very nicely. The yellowish is ice, water ice, and the blue is the protein. And interestingly, the protein can connect to the A plane of the ice crystal. This is an ice crystal in typical growth form, this hexagonal ice. And usually, if there are no proteins around, the ice will grow undefined in any directions. It's a so-called dendritic growth. If you freeze water under the microscope, you see dendrites, little teeth, growing suddenly in all directions from a nucleus, completely uncontrolled, and an alga being might be damaged by that. But the ice structure in protein connects to the A plane of the ice crystal and blocks it there. So the ice crystal can't grow into the A direction, only in the C direction. And what happens then is that this crystal here, this is a water crystal, and you see this hexagon in the middle and the pyramids on both sides. So it's a hexagonal bipyramid, it's called. The protein is blocking here on the sides and blocks it to grow in the horizontal phase now, and the ice crystal can only grow up and down. So it forms this little pyramids, but it's a controlled growth. It's not uncontrolled growth in a big crystal, but it makes the crystals stay small and so less damaging. That's a hypothesis behind it. There's a lot of hypotheses. There's a separate group dealing only with these ice structuring proteins or antifreeze proteins. There are cute people and great people doing this research, a very different and very complicated system research topic. Here on the top right, you see such a crystal. This is artificially colored in yellow to make it better visible. Under the microscope, this is a nanoliter osmometer apparatus where you can freeze water with a protein. So it's no algae involved anymore, just a pure protein solution being put in a very, very tiny little hole in this nanoliter osmometer. And there's a very precise temperature control on this device. And you can produce one single ice crystal, and then you can manipulate the temperature very slightly up to increase the temperature. And then you see how this minute ice crystal is growing. And you see this on the bottom right. I hope the video works. You see this little minute ice crystal here. The size is approximately like 50 or 10, 20 micrometers. And if I start the video now, you should see how the ice crystal growth influenced by the protein. It's not uncontrolled, but very controlled. I hope you see the video now. You see how the crystal growth, and on the surface, it's, might start it again, yeah. You see how each layer of ice is growing on this crystal in a very defined and controlled manner. Interestingly, we asked ourselves, and also the other AFP colleagues asked themselves, where did these proteins actually came from, and where did them evolve? And interestingly, if you look at ice binding proteins or antifreeze proteins or ice structuring proteins from the insects or from the plants, from the higher plants, or from the algae, from marine algae or freshwater algae, the mode of action is always the same. You will always observe this ice structuring modification of water crystals or water ice, but the homologies on nucleotide level are very different. And also on tertiary level, if you look at the proteins and the amino acid, they are very different. You hardly find homologies only in certain motifs. And if you look back and combine these motifs, you find homologies also to antifungal proteins and to endochitinases. So the endochitinases, obviously enzymes which can dissolve chitin. Chitin is what the insects surround. And also antifungal fungi, also the cell wall of fungi also is composed of chitin. So it could be that what we follow a little bit this idea is these ice structuring proteins might have also an activity in destroying chitin and maybe have an antifungal activity, which would be ideal to develop, for example, a new antifungal pharmaceutical instead of those we are looking for at the moment. We are all facing a lot of antibiotics not working anymore, antifungals not working anymore, and having such natural antifungal substances would be very interesting. But yeah, it's very hard to follow this line because, of course, during evolution, these proteins have evolved a lot. And so far, we didn't observe any antifungal activity, at least not in the ISPs we have found. Originally, if you look into genomics and transcriptomics, you find that these proteins obviously have been developed or evolved in the very beginning in bacteria, and then being transferred to higher organisms by horizontal gene transfer. So it's not the algae which have found these proteins, evolved these proteins, but they have taken them up from bacteria. In our strain 50, you might guess it already, we've studied a lot and very intense from different sites, and currently a student at my lab finished his bachelor thesis, and he also looked at the sequences. These are sequences from earlier work by other students, but the student now, I will present a slide later, but we could detect the amino acid sequence of this protein. And what is obvious, and what also these motifs I marked here in blue squares, these motifs are consisting of threonine, a different amino acid, and then a different another threonine. So these so-called TXT motifs, and these are presented on the beta strands of the molecule. You see this beta strands here on the top part of the protein, and here on the model on the lower, below that, these blue ones are the threonine rests and residues. And on this beta strand on this side, this is actually where the protein binds to the ice crystal. That's the way where the ice crystal, the theory behind it is this distance between the hydrogen molecules of the water molecules, hydrogen atoms, so the two H's of the water molecule, the H2O, has a similar distance like the distance between the two threonines on these beta strands. And obviously, this enables the protein to bind to the water crystal. This is very, very, very strange. That's the hypothesis at the moment, but I'm not an expert in ISP, but a very interesting field. Interestingly, in all organisms, these proteins are not secreted into the cell, but it's excreted into the extracellular compartment. So in the blood of the fish, into the hemolymph of the insects, also in the algae, it's excreted into the cell medium. We can harvest it easily from the culture medium, and in higher plants, it's excreted in the apoplastic space between the plant cells, where it obviously has its action part. The effect, as I said, it's not lowering the freezing point very much, but it can modify the ice crystal in its habitat. So the ice crystal habitat is modified. The function, as I said, we assume it's dispersal of the algae with the wind to make to disperse the algae wherever they want to go. On the small scale, in the microenvironment, it makes water available to the algae because the algae are always connected to the ice crystal and get in their cell close environment this liquid water which they need for photosynthesis and for the metabolism. For an industrial application, it's not really clear. To assess the activity of such proteins, there's a so-called recrystallization inhibition assay, the RI assay, which is easy to perform under a cryomicroscope. What you basically do is you freeze your ISP solution. Here it's a culture medium, not even the purified ISP, but the culture medium, just concentrated. This is a picture what it looks like if you freeze the pure culture medium without the ISP in sucrose. You add the sucrose only to obtain these small crystals you see here. And if you now wait for an hour at -5 degrees under the microscope, what you observe usually is recrystallization that happens due to physical energy conditions. If you have small curvatures of the ice crystal like you have here, the curvatures of the ice crystal are very small. And in physics, it's high energy areas, and high energy means that they tend to gain enthalpy. They tend to make lower curvatures, like here. Lower curvature means lower energy, and so the enthalpy is lost. And so what basically that happens also in your frozen food or frozen fruits, frozen meat you put in your freezer when you buy it. After a while, you get bigger crystals, and your ice cream also becomes less smoothie. It becomes a little sandy on your tongue. You basically taste these bigger crystals. This is due to the recrystallization process happening to everything which you store at not so freezing temperatures, when crystals can still transform to larger crystals. Now, if you put ISP in the tests, and here again, it's only the culture the culture supernatant, concentrated, but not with a purified ISP. We can calculate it, and we know in this culture medium we have about 2 microgram ISP. If you then wait for 1 hour at -5 degrees, you see a slight increase in the crystal size, but not as much as before. So this is a typical activity of the ice structuring protein. It can make crystals small, even under ungood conditions for your freezing products. And also you see the form is still not so roundish as before. Here it's very round. And here the crystals stay spiny, little, not so roundish. And this is from, again, this microglia Clinobiasis. As I said, applications in industry, it's very hard to find something. In food industry, Unilever produced in the United States of America and North America, they brought out on the market this Breyer's ice cream, it's called. They put antifreeze protein from an Antarctic fish in there. They produced this antifreeze protein transgenetically in a yeast, and then put this transgenetically produced antifreeze protein in their ice cream, because they wanted to take out of the fat, because fat in ice cream usually makes this very smooth texture of the ice cream, and that's what you want to have when you eat the ice cream. It's very smooth and creamy. But if you want to produce low-fat ice cream for diet, or for people who don't want to eat so much fat, of course, you have low-fat ice cream. But if you take the fat out of the ice cream, it gets the sandy taste on your tongue, and you don't want that. So they put in this genetically fish protein in their ice cream, and that ice cream was still smooth on the tongue. That was only produced for the North American market, as I know. It never entered the European market, because on the European market, transgenetically products are not allowed, especially also not on the German market. The customers don't accept it. It's obviously different in the North American market, but here the market rules the product. If the people don't want it, they won't buy it. So this product never entered the market. This is the only product I know of worldwide where antifreeze protein ever were put into a food product. Also, we tested it on meat. We froze meat with and without our proteins, and there was no really advantage. So we thought that this freezing damage from freezing meat, when the liquid comes out of the meat cells, would be decreased, but that doesn't work really. As I said, this genetically fish protein, having it in ice cream, is not really nicely marketable. I think Unilever never told the people, and you don't find it on the box, that it's coming from Antarctic fish. It's not as good sound having antifreeze protein fish in your ice cream. You don't want to eat that really. We thought of maybe using it in the agro industry to increase cold tolerance in crop plants, but it's very problematic in the European Union. It's not allowed there, so it's not marketable in medicine. We tried it also because, of course, a lot of people said, hey, you've got snow algae and they are cryophilic, so they should be easily cryopreservable in liquid nitrogen. But of course, living in frozen glacier is a completely different condition than being frozen at minus 150 degrees. As I said earlier, in the glacier, when it's covered by snow in winter, even in the polar regions with air temperatures of minus 29 degrees or minus 30 degrees in winter, under the snow protecting cover, you have -7 degrees only. And -7 degrees, you can easily avoid freezing with sugars and other glycerols, for example, glycerol and proline, which all the algae produce to prevent freezing. So in medical applications, it's maybe could be used for treatment hypothermia, protecting cells. We don't know really. Cryosurgery, possibly. A lot of surgeries made with frozen sons, frozen in a liquid nitrogen to kill certain areas in your tissue, and you don't want to kill the surrounding cells. Maybe they could be treated with these antifreeze protein solutions to prevent damages. We don't know. That's only ideas. Also, maybe even adding ice structuring or antifreeze proteins to transplants. If you have kidneys used for transplants, you know the whole world is lacking suitable transplant organs from patients who have been dying, being transplanted in people in patients who need kidneys or heart transplants, and we are short of these organs, of course, and they are being transported in being perfused, as it is called. So when they are transported around the earth to be flown into hospitals for transplant surgery, they are being perfused with a hypothermal perfusion solution, which keeps the organ alive during transport. And maybe one could lower the transport temperature by a few degrees, making the transplant organs living for maybe 1 or 2 hours longer, which equals to having more transplant organs available on the world. But this is medical research and very, very complicated. Of course, performing these studies is very complicated as well. So it would be interesting, but hard to perform. Another Fraunhofer Institute here in Germany, as I said, we've got 70 institute, and the IFAM in Germany also made studies where they put antifreeze protein. You can buy, not really anymore, but you could buy it at that time, and put it into antifreeze paint, which is applied to airplanes, so that in winter conditions, when here in Europe you have winter conditions, and the planes have to be defrozen on the outside with antifreeze solutions before takeoff, not to freeze during winter times. But if you look at the costs for putting such valuable proteins into paint, or even solutions being sprayed all over an airplane, you can imagine how expensive that would be, and compared to chemicals being applied to planes. This is a high imbalance of the costs involved, and this is something you, to my opinion, would never get on the market, just because the cost is far too high. Switching a bit into the genomics, a few years ago we started with colleagues from the UCLA, the University of California in Los Angeles and Berkeley. And in Berkeley, I must say, the lab moved from Los Angeles to San Francisco, I think. The Merchant Lab from Sabir Merchant, and also with the Benning Lab here at the GFZ, the Geological Research Center in Potsdam. We started sequencing the genomes a little bit of some algae. We observed some very strange things. We all observed that the genomes were very, very, very high. When you sequence Chlamydomonas reinhardii, we know the whole genome consists of about 100 megabytes in nucleotides. When we sequenced the genomes of the psychophilic algae, we observed a fourfold or in eightfold even in the genome size. And people asked me, why is that? Is the alga diploid or polyploid? And I said, no, all I know from my knowledge, our algae usually grow in the haploid stages and the vegetative stages. One single cell divides into 2 cells, and it's always haploid. And when they form zygotes, it's diploid, but shortly after, they divide again with meiosis, and then you have the haploid stage again. So I would expect a genome size like Reinhardt-E. And then after a while, I also read and heard from other colleagues that also a lot of other polar species tend to have diploid or even octoploid genome sizes. And we assume that is actually just to make sure that all the enzymes are located and all the proteins which are needed for life in this extreme habitat are available on different positions in the genome. And during when the transcriptome is being put up to produce the enzymes, then the machinery, the molecular machinery, has different positions where it can process the tRNA and the mRNA and the tRNA, and then produce the enzymes. So it's a more transcripts are available to produce the special enzymes the alga needs to survive the freezing temperature. That's the explanation we have at the moment. But on the other side, also Rhaphidonemma, which is also psychophilic, but from a completely different family. It's from the Terbruxophyceae. These Chlamydomonas are from the Chlamydomonaceae, and the sister class is Rhaphidonemma, the Terbruxophyceae, where a lot of soil algae are in there, and also all the Rhaphidonemma species from the glaciers, which are also psychophilic, and they have a very small genome size. And the soil algae, also cryophilic or cryotrophic in some parts, have a similar genome size than Chlamydomonas reinhardtii. So different strategies, again, in these psychophilic algae from the Chlamydomonas and Chloromonas groups. The problems, as I said, first it's problems. Later on, when you know it's a problem, you know how to deal with the problem. But we observe polyploidy in psychophilic species, high increase in the genome size. And another problem we have currently is the de novo assembly of the genomes and the transcriptomes because we have missing templates. It's just a tedious work to have annotations. And that keeps us busy over the last years already. We're slowly getting our results from there, but I'm not a molecular group really, but we do this a lot with incorporations. We have done a lot of studies and experiments. We exposed our two different species, Chloromonas nivalis. I didn't change the name here. It's Chloromonas remiazii, the strain number 5, and strain number 1599. We exposed it to sodium stress and to CO2 stress to see how the CO2 uptake mechanism is maybe different from normal algae. Because it's also decisive, of course, to live in the cold. And we have put it to blue light stress and red light stress because we know from higher plants that blue light receptors exist also in microalgae. And the blue light receptors, they measure basically the quality of the light, not the quantity, but blue light penetrates earliest in the snowpack. So possibly a blue light receptor could signal the cell at the beginning of the summer, hey, it's summer coming, because the first light is penetrating the snowpack, is reaching the cell, and maybe this activates the life phase of the cysts, okay, or the spores, because these green stages don't exist possibly during the winter phase, but a resting stage, and the blue light maybe kind of signals the germinating process, we assume. But we are only at the beginning of sequencing these transcriptome samples. And red light, we also know from higher plants that red light has an impact on the physiology. So that's why we do this for stress experiments, for comparative studies later on. And in the soil algae, in this red-green system, which is very easy, as I said before, low nitrate makes the red cysts, high nitrate makes the green cysts. Is this very special? What are the mechanisms on molecular levels? So we did transcriptome samples on these, and they are also being sequenced currently. It's a long way, I can tell you. To understand if the cold active enzymes are interesting at all, this is data from 2007, very, very, very early studies when we didn't do any transcriptomics so far. At that time, they was quite expensive, but a student of mine, he sequenced the Maillard dehydrogenase, the MDH. This is an enzyme from the Krebs cycle, so it's involved in the energy physiology, energy metabolism of the alga, a very fundamental metabolism, of course. And we compared in an enzyme assay the activation energy of the transcripts or of the enzymes from Reinhardii, which is the orange curve here, and from several cryophilic or cryotrophic algae. And here you see in the diagram the temperature of the activity or the relative activity of the enzyme of the different organisms, and we observed the maximum activity, the temperature of the maximum activity of that enzyme is shifted to lower temperatures by 5 degrees Kelvin or 5 degrees or 10 degrees Kelvin even, to lower temperatures in cryophilic algae. In the psychophilic ones, in our strain 20 in this case, it was investigated, it was shifted by 10 Kelvin to lower temperatures, and in others by 5 Kelvin. And even the small shift in enzyme activity, if you translate it to industrial processes, if you imagine, for example, any kind of enzymatic process to process biomass in whatever food process, if you can lower the temperature in this process by 5 degrees or 10 degrees, you can imagine that that serves a lot of energy in the industrial process, and that saves a lot of costs in the process. So the production or the development of cold active enzymes and the employment of these cold active enzymes in food processes in the industry could save a lot of energy and could be very advantageous for such companies. We are still not that far for a product. This is from a bachelor thesis who just was finished by Fabian Dressler here in my lab, and he also looked for a start at the amylase of our strain 50 and compared it with other amylases from Reinhardii. And what he observed is that the sequences themselves are very similar, but only 70% are homolog to the amylases on nucleotide level to Reinhardii. They look very similar on tertiary level. If you look at the amino acid sequence, they are very similar. But if you model the cold active amylase from that strain and compare to Reinhardii, you observe modifications in the catalytic center. So the catalytic center is where the substrate, in this case starch, which would be lysed through the amylase, is binding, and you have less in the whole structure of the molecule, you have less intramolecular bonds, and that makes the whole molecule of the enzyme more flexible. So the catalytic center and the binding regions of the molecule are more flexible, and so the binding to the substrate, to the starch, is facilitated. And so in cold conditions, due to the higher flexibility of this enzyme, it has also increased binding capacity to the substrate. That's all the general thinking, and Collins and Margeson, they published it in this publication down here, if you want to read it. So the advantage on a product level is you can use lower energy, lower costs, lower heating if you involve a cold active enzyme compared to a normal enzyme assay that works at room temperature. I don't know if you have it in South America, but here we have from Ariel, from the washing detergents, we have it's called Ariel Cold Active that was developed, I guess, 15, 20 years ago. And they put in a psychophilic lipase, I think, from a bacterium, from a psychophilic bacterium, to have a fat soluble enzyme in there, which is cold active. And actually, this cold active Ariel detergent you can use at 15 degree or at room temperature, and it takes out your lipid, your fat stains from your clothes at this low temperature, so you don't have to clean your clothes at 40 degrees or 60 degrees. And this is very good, of course, because doing a laundry at 60 degrees or doing your laundry at room temperature saves a lot of energy for yourself, for your household, but also worldwide, of course, for all mankind. So the use of cold enzymes is quite interesting. Another example might be if you want to, for example, get olive oil out of olives, you have to process the olives and you have to macerate them, you have to tear up all the tissues, and if you have a carbohydrate you can put in there, and you want to save the olive oil from decomposing at higher temperatures, which would be good for normal carbohydrates. But if you have a cold active carbohydrates, you can macerate the olive tissue and gain more olive oil from your olives. And again, you have a higher extraction level of your olive oil and you have more to sell. You have a higher gain from your products you sell due to cold active enzymes. So there's a good market for that. That's why we look into all the transcriptomics and especially in the enzymes. On the one hand, you have cold active enzymes, proteins whatsoever. It could be used in crop resistance if you would make transgenic crops. Could be a resolution to make such products. Cold protective cosmetics is very special. High product cosmetics is a good market because you can sell such products at very high costs. On the other hand, we have high costs in the production of the algae, and that is the only way you can basically justify why to put in such very cost-intensive products from algae. Otherwise, it's money ruling the market, really. The light adaptation, as I said, we haven't found them, but you might also put photolyases into light protective cosmetics, where your cosmetics might be able to reduce damages from radicals on your skin. So you have maybe your photolyases in your skin creams. Don't ask me or question me if it's working or not. Cosmetics sell on the market. I also think the South American market for cosmetics is really big. The European market is big. The Asian market is huge. And again, all the cosmetics market lives from trends. The cosmetic companies want new trends, new products. And currently we follow that up, and we have our industrial cooperation with that as well. Molecular involvement in cosmetics is very fashy at the moment, very trendy. If a cosmetic ingredient is molecular, then people tend to believe that these cosmetics are also effective. I mentioned the field of optogenetics. This is very special. One of my colleagues here at the Humboldt University just gained a research prize for that, a very well-renowned one. I don't remember the name currently, but he's the father of optogenetics. What you do in optogenetics is you use these blue light photoreceptors. That's why we are so keen on finding different blue light receptors in the algae. With these blue light receptors, they act as proteins in the membranes, and they can be switched on and switched off with blue light. And you can use these channels to activate, for example, a potassium influx in the cell. And you know that potassium is active in activating a lot of cell processes. So you could actually, for example, in medical applications, include such a blue light receptor in cells, incorporated, for example, in the brain tissue maybe of a human being, and then switch on this channel with blue light. You can apply from external, because blue light will easily penetrate the skin, and then you can activate such a cellular channel to activate a potassium influx or whatever to start a medical treatment. Very medical, the whole thing, but a lot of people working on this subject. Aridness, I told you there's a lot of desiccation in these snowfield environments. Again, we have desiccation problems or in crops, desiccation around the globe. Possibly crops could also be made more desiccation resistant. Same again for cosmetics. Maybe you can put these products the algae produce to avoid damage from desiccation. These EPSs, for example, in cosmetics. So these cosmetics retain the humidity of the skin, retain the water from the skin instead of letting it out of the skin cells or from the skin tissue. And in general, of course, the extremophiles never doesn't mind if they are from cold extremes or from salt extremes or alkaline extremes, or you have algae growing at pH 2, for example, which of course are interesting, or from hot environments. A lot of people do extremophilic research to involve or put these ideas into products for food processing or for diagnostics. Currently, I think all these products are only realizable really in high-priced markets, for example, in medical cosmetics, in medicine, or in diagnostics, or in cosmetics in general, because these markets, the products are themselves very expensive, and so you can put also expensive raw products in there. And with that, I'm coming to the last point. I think we are easy in time. I don't have a clock here at the moment, but this is only very short. This is pointing at the products we have on the market. This is just to tell you again that the transformation of our industry, in Germany at least, we have since years having a transformation of the whole industry from petroleum-based raw materials, from traditional raw materials into natural materials. So ideally, the algae should save the world from what we did to it during the last hundreds of years from industrial evolution. Of course, the microalgae won't save the world. It's only the human being who can do that. We, in my opinion, have to consume much, much less energy to survive on our Earth. But of course, we do our best to replace traditional products with algae-based products. Easiest way is omega-3 fatty acids. I told you, so far it's extracted from fish scrap or bycatch. Fish we don't use for eating, but we only take the oil out. In future, it could be marine microalgae or permafrost algae or snow algae, or also the Labyrinthomycetes. Some call them under the algae as well. But these are actually kind of fungi which also produce these EPA PUFAs or DHA PUFAs very nicely. Astaxanthin, this dark red pigment, it's currently also produced in corn and tobacco and yeast. Wrote this wrongly here, yeast, but it's transgenetically. Also some algae produced at Hematococcus. Currently it's being produced on Hawaii in large amounts, and it's being used for aquaculture food to produce all the salmon we want to eat, and so that it looks so nicely. But also, as I told you and as I showed you, some of the permafrost algae also produce astaxanthin, and we also work in developing new foods for aquaculture systems. Beta-carotene, it's produced transgenetically in algae also already, in Dunaliella, Salina, in Israel in huge amounts, so there's no need to develop a new product system. They do it industrially, and it's very nicely, and there's no way to enter this market with cryophilic algae. Lutein is the same. Our algae produce it, but so far it's being produced, and also in the future, I think, in Tagetes, the genus marigold, it's called in India, in huge amount. If you see the Bollywood movies from India, they throw around, and every party is with a lot of marigold flowers, petals, and all these marigold, this yellow color comes from lutein. And they produce it in such huge amount and serving the world with lutein. Thickeners, again, so far it's petroleum-based, microplastic, it's all petroleum-based. This could be replaced maybe in future, and it has to be replaced really because we don't have that anymore, and microplastic is really bad for us. So there should be an urgent need to develop natural EPSs or foamers or thickeners which can be decomposed easily. Pigments for cosmetics, all the blues and violet and orange and silver and metal pigments you find in your cosmetics, they are all mineral based. They are all coming from mines which are not environmentally friendly really. A lot of child employment is there, so it's society is very bad, and it's an urgent shift to natural pigments, and they exist. Cyanobacteria pigments, the blues exist, and they are to certain amounts also incorporated already, but also the other pigments can be replaced with natural and non-hazardous natural pigments in cosmetics really, at a higher price. That's always what's written here on the right side is higher price, and the question is always, is the customer willing to pay for it? Pigments in food, it used to be very synthetic. It changed a lot over the years. A lot of pigments are coming from fruits and plants, and also cyanobacteria, and will be also in future. But the question is always for what price, and is the customer willing to pay the price? As I said, we produce some metabolites for cosmetics because it's a high price market, and we develop these photobioreactors. This is Felix Jorde, former engineer, and he grows the algae in a very, very high quality, highly pure, with no bacteria in there, because our customer doesn't want any bacteria in there, which is not easy in higher volumes. But we can do that over the years. That's being done by over 10 years now. And we serve this cosmetic market at a very good price. And the cosmetic company also gets a good price from that. This company also can have that advertisement because they have innovation prizes, and it's very environmentally, it's ecologically, and that's also where the cosmetic counts on. It must be innovative, it must be environmentally friendly, it must be natural, it must be ecologically, it must be certified, and then you can launch a new product. And they claim from our algae, if you Google snow algae powder, then you will come up with all the products going back to our algae. We are not mentioned anywhere. It's only this company, Mibel. They are a company in Switzerland, and they sell their raw product to companies like Bayer-Store, for example, and they sell it under the brand name La Prairie, which is a very, 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 very high price luxury cosmetic on the market. And now also by lower price market brands, La Mer and Dr. Grundle and so on. But they've been successful on the market over the last 10 years, but only because the cosmetics themselves are very, very expensive. Yeah, and with this, I'm at the end of my talk. I have to thank a lot of people, all my cooperational partners here in Canada, for example, from the Quamby Lab, with whom I did a lot of taxonomy and phylogeny, the Merchant Lab from Sabea Merchant in the USA, where I do a lot of genomics and transcriptomics. Same with the Benning Lab in Potsdam here, next door, with the German aerospace company, society, and the Mayo Clinic in the USA, where we do the analysis of the ISS experiments and the mutation analysis. Also with Czech colleagues, we do a lot of taxonomy, and of course a lot of people from my lab who did all these studies over the years. I also like to point you to our snow algae meeting. We established this in 2016, I believe. Normally we do it every 2 years, and now last year actually would have been the third, but we had to move it. We did a virtual meeting, and now we want to do it next year in Prague, the fourth meeting. And if you're interested in pursuing the snow algae community, we're growing from 20 to 60 people now. A lot of people around the world meet there, very informal meeting. and you're welcome to connect connect to me, and then I can put you on the email list. And with that, I'd like to thank you a lot for staying on the screen. I think it was a long talk. The 3 hours are really hard to stand if you're just listening and listening and listening. I hope I was informative and interesting. It
was perfect. Very, very, very good. We have some questions here. Can I put in the screen?
Sure. So Vanessa asked, we know that algae are an important source of bioproduct. However, according to experience, how could we make the link between research and industry? Yeah, I mean, I had it easy because when I moved to Fraunhofer, I had to learn how to talk to industry. And I think the scientist at university who's doing basic research doesn't know how to talk to industry. And you just have to step forward and get in contact with them. And then you will learn what they asked for. They have very different pre-requirements. The first thing in cosmetics, the first thing that they asked you, do you have the rights on the algae? If you don't have the rights on the algae, they will never put up a patent. And then they don't talk to you anymore, for example. Or, of course, the question is, can you produce masses of that? When we can produce all algae in the lab in these small volumes, like 100 milligrams for genetic analysis, but the industry needs 100 kilos, or maybe a ton, or even more if you want to replace really the traditional products. So just approach the industry, try to get in contact with them, and go on the congresses where industry goes and talk to them and try to understand each other. The industry needs us, and I think we need industry. It's a long way, but start understanding them. That's, I think, the most important thing.
Good.
Is there potential for biofuel production from this microalgae? No. In my opinion, very easily, no. We had research programs in Germany over the last 20 years, I think, was always bio-kerosene, biodiesel for airplanes and for cars. They had a machinery up in Hamburg, for example, in northern Germany, and they had a production plant there to produce bio-kerosene for an airplane. And they want to establish a regular flight to Munich, which is in the southern part of Germany, only 700 kilometers away. And they want to establish a regular flight once a week based with biokerosene from algae produced in Hamburg. And that airplane doesn't fly anymore. And I know some pilots who said from America, and I remember him saying, there is no way no one's going to get biokerosene from algae in my little plane. I'm counting on the good kerosene from fossil energies, and I can understand it. And I mean, if you calculate the amount of masses of kerosene we waste on flying from here to there, and a lot of people in Europe, Berlin, doesn't need to tell you, a lot of young people fly to Berlin for the weekend. A lot of Berlin people fly to Barcelona for the weekend just to have some fun. We can't afford this anymore. So clearly, forget about that. Use less energy. We will never be able to produce so much bio whatever to drive our cars or fly planes. No way, in my opinion.
Another question from Vanessa.
What is it like to connect in such remote? What is it like to connect in such remote places? Do you have a funny story to tell? Any difficulties that the group experienced? In Brazil, we call these situations perengue. I don't know what perengue means.
Perrengue é a palavra.
What is in English?
I don't know. It's like problems. It's like problems. But it's a funny word here in Brazil. Perrengue.
Well, it's funny if you're the captain on board and looking out and you're safe on the board, it may be funny. But if you're running around in your polar suits with all your bags collecting samples and your esky on the one hand and your coat and you're sweating because you're running around, and then suddenly he calls on the walkie-talkie and says, "Please come back to the ship. There are 4 polar bears around." then it's not funny. Then you try to grab your weapon, you have weapons with you, and you look around, you don't see those polar bears because it's really, you're in the middle of nowhere. It's like you being in the jungle of the Amazonas. You don't see the jaguar coming around the area. You're just fucking frightened that the polar bear will come and eat you. And on Spitsbergen, the polar bears attack human beings because they are just food. They are not afraid of people. So it's not funny. It's great. I mean, I thank my PhD advisor from university, Professor Fuhr, for the remarkable 20 years I had and for the 7 expeditions I did to Antarctica. I will never forget that. And it was funny in a way. And living with 4 men on a yacht after 6 weeks, you don't smell each other, maybe because there are not many bacteria around in the polar environment, but there's no way to shower. So you use the ice cold water from the Arctic Ocean, running in for 2 seconds, being frozen instantly, and then running out again. Life in the polar regions is funny in expedition. I have a lot of funny stories, even from our administration, understanding my research work. They don't understand a way. In Munich, our administration doesn't understand what expedition means at all. But that would last until 8 o'clock if I tell you those stories. Very funny. Funny in a way and funny from the other side.
Yeah. I was talking with some colleagues that is a spaceship work because here in Brazil, for example, who can say that, well, I was running for a polar bear? Maybe for a wolf, for a snake, but never about a polar bear.
No, there's no way. Because if you see them really, I mean, first you have to spot the polar bear, and you're in an area where it's all snow around and rocks, so you don't spot the polar bear before it's like a few hundred meters away from you. We had this. We were very close we saw a polar bear very, very far away, and we could monitor it. And suddenly, I mean, you don't look at it for only a few seconds, and then you see it's so close suddenly, and it can speed up so fast. You would have no chance to run away from him. So the only way is to shoot at him with very strong weapons. Really, you are compulsory to wear weapons, or the signal pistols that you shoot in the snow so that he is getting afraid of you. But he's not afraid of human being. And he's also in water. He can swim to your zodiac and give your zodiac one slap with his paw, and he will sink and he will eat you. So it's no funny way to fuck around with polar bears. No way. Just get away. It's a bit.
No, I have some doubt also. Well, I work with diatoms, and until we know the division cycle of diatoms, the synthesis of the proteins are made during the S phase, and S phase occurs during the night, during the period that we don't have light.
Yeah.
Well, I was thinking about algae that live in extreme north and extreme south, because we have, for example, some periods that we have the day without night, or some days without sun.
Yeah.
For example.
Yeah, this is very interesting, really. I mean, that's one thing I mean, that's why we basically collected the samples. Actually, I also have some diatoms from snow from Antarctica. If you're interested, you should work with them. They are from Antigua's base. There was a completely brown snowfield, really freshwater diatoms on snow. And we have 2 or 3 different species. And it's I don't know how they can perform all these physiological things they normally do during the night, being exposed 24 hours per day. There must be, of course, a different machinery going on, and that's part of the transcriptomics research we could do. I have no clue. I don't have an answer. They seem to be doing everything very efficient during the day. I mean, even in basically from I would say July is only 3 months where you really have complete daylight. From June, July till August, like mid-August already the sun sets a little. But if you really need a dark cycle, no light, they must have shifted it also into the light cycle somehow, or excluded the problem of having light around.
Okay.
Interesting.
We have a last question, and after some comments that I would like to put here in the screen.
Yeah. Jose asks, what is the perspective for industrial scale production of these microalgae products, perhaps because of transgenic organisms that we are already able to cultivate with greater ease? Yeah, that's what I said. The German market, people in Germany are green, and I myself am green as well. I don't necessarily want to eat transgenetically produced food. In different markets, it might be different. It might be easier on the American market, in the North American market, on the Asian market, I don't know. But for the moment, if you want to introduce natural algae products, it's very hard, and only on the trendy markets of novel foods and so on, you might get in there. But then you have to obey the novel food regulation and so on. So there's a lot of industrial regulations coming up as well. I agree completely as far as if you produce a metabolite only, which you extract later on, like astaxanthin or even medical products. I mean, everybody knows that insulin or whatever is produced transgenetically, and everybody who's dependent on insulin will never say, I'm not taking it because it's transgenetically. If you take out the product after you've produced it in a transgenic organism, I would feel better with it. I just wouldn't want to eat transgenetically manipulated corn, for example, because there is more carotene in it or whatever. Then that's not the right way to do it. I'm too green for that. I'm too eco. It's a big problem, yeah. But yeah, it's a price thing. I mean, natural products cost more, and in 20 years it might be different when there's no fossil fuel anymore, and it costs maybe a barrel costs $1,000, not $100. Then people might think differently. If they have to pay simply more or don't get it anymore, a product, then they might eventually pay more. The market will change, definitely.
Okay. Then we have 2 comments.
This is very nice. Thank you. Good to hear. Yeah. Whenever you, whoever wants more information, just approach me via my email. If you Google, as I said, CC Cryo, you get a link, and you will find my email address. Go for it if you're interested in corporations or in strains. I can advise you whatever ideas you have, or come over for work for here. I'm happy.
Do you accept postdoctorate students?
Well, postdocs, of course, if I have the money, that's the problem. Master students and bachelor students are cheap, and we accept them, of course, easily. PhD students or postdocs, if I would have money, postdocs, we can't afford it. Nowhere in the institute currently, I'm afraid. But if somebody comes up with a project together, I'm happy. I'm open for any cooperation, of course.
Great. Then I would like to thank you so much your participation here.
My pleasure.
As said Emily, it was a very great, amazing moment that we learned a lot. Then I would like to invite you to participate with us with the other lectures if you want.
Yes.
If you want to see. And the next events, the next editions of our course, if you are available, you are invited again. Thank you so much, Thomas.
Thank you very much. It was my pleasure, really. It was very nice.
Okay, if you want to say the last words here.
Well, it was just a pleasure, and I thank you very, very much to stay so long on screen. I know how hard it is. I'm not a friend of this virtual meetings. I would have loved to come over to Londrina and hold the talk there, or maybe come to South America in general for whatever meeting, and maybe I meet whomever by you, maybe in person somewhere. feel free to approach me, and it was a pleasure for me to talk to you.
Ok, thank you. Então, pessoal, encerramos mais uma das nossas transmissões. Fiquem bem, até mais. E à tarde a gente tem a participação do professor Orlando Neck Jr. falando sobre Rhodoptera, certo? Até mais, pessoal.
Bye-bye. Wellington, you sent me the